Good afternoon, everyone. My name is Fengxiao and goes by Bu. I'm a PhD candidate working in the genetic aspect of the aHUS. Today, I'm going to give you a talk about the current research of the aHUS that happens in this lab and as well of the world. So as we discussed this morning, the aHUS is caused by the dysregulation of complement pathway on the cell surface level. In healthy people, the C3 convertase automatically generated and deposits on the cell surface. But in healthy people, they have those regulators, complement regulators, to stop the activity of the C3 convertase. However, in the aHUS patients, because of genetic or acquired defect in those complement regulators, the C3 convertase start working, cuts more C3 to C3b, and generate more C3 convertase, forms the amplification loop. In turn, it generates the C5 convertase, and the C5 convertase cuts the C5, activates the C5, and generate it's called the membrane attack complex, which then destroys the cell. Then what happens next? So we all know that if the blood vessel is broken, you will bleed. And to stop bleeding, there is a process called coagulation will happen. So basically, the coagulation system will generate those blood clots on the damaged area to stop the bleeding. It happens in our body, of course, including the kidney. And this is a schematic picture of the capillary, those very tiny, small vessels in our kidney. It's very small. The vessel wall is formed by a single layer of the cells. And the capillary is so small that it only allows one red blood cell to pass by at each time. So what happens in the aHUS patients is that because of the overactivation of the complement, numerous cells are damaged in the blood, on the blood wall. So it forms a lot of blood clots. This step consumes the platelets. That's why you see the platelets number drops down in the patients. And when the red blood cell passes by, it has to squeeze through those blood clots, and sometimes it breaks. And that's why you see those schistocytes or hemolytic anemia in those patients. So clearly, as we discussed, the coagulation pathway is also involved in aHUS. Dr. Smith discussed these two papers, and there are other publications that reported, for example, the thrombomodulin gene is involved in the disease. And there's a group tested the gene expression, those coagulation gene expression is changed in the kidney of the patients. So given those evidence, we decide to screen all the genes in the complement and the coagulation pathway. We developed a panel called CasCADE, used the next generation sequencing techniques to screen 85 genes in the patients who has the aHUS. And another version of the CasCADE, we call it the TMA, is kind of like a smaller version. Now we are using it for the clinical genetic test. I used this panel, screened 36 European American patients. Consistent with previous report, we found that factor H is the most frequently mutated gene. That makes sense. Surprisingly, the second most one is plasminogen. Plasminogen is the most important enzyme that removes the blood clots. The mutation in this gene will slow down the removal of blood clots. So we believe that those mutations in the plasminogen will modify the aHUS symptoms. The paper has been published last year. And based on the results and other papers, I generated a modified disease model. So start with the trigger events. The patients have the genetic defects in their complement system. So their complement system is overactivated. It will damage the endothelial cells, or you can imagine it damaging the blood vessel wall. And the patients should also have some defects in their coagulation pathway. Together, they will generate a lot of thrombi, those blood clots that block the blood vessel. And those thrombi will destroy the red blood cell. It will release a chemical compound called heme. The heme is a secondary activator of the complement pathway. So you can imagine there is a dysregulation loop going on. And with this dysregulation loop, finally the patients get the onset of the aHUS. A great example of this new model comes from a aHUS pedigree. So there are two kids got the disease. Their biological mother and grandmother also carrying the FACT-H disease mutation. However, the two kids got the disease from very young age, and their mother and grandmother never got the disease. So we screened this pedigree using the CasCADE panel, and we find that the mother and grandmother also carrying a mutation in the factor 10. Factor 10 is a coagulation gene. It promotes the formation of the blood clots. And what does this variant do? This variant has been reported to cause factor 10 deficiency. That means if people have this mutation, they will have a lower level of the coagulation activity. So we did a computational analysis to see what could this variant do. And if it turns out that this variant actually changed the factor 10 structure and the protein level. Then we also did a bunch of experiments, and we found that this variant stops the factor 10 secretion into the blood system, into the bloodstream. So we found that in the mother, she only has half amount of the factor 10 comparing to normal people. So there are more questions to be answered. Why we want to know how exactly does the protective variant work? We do have a hypothesis. Because the mother and the grandmother carry only one copy of the normal factor 10, they can have only half of the factor 10 comparing to the normal people. So it's less likely for them to form the thrombi. So we are not going to that dysregulation loop. They may do have the overactivation of the complement, but they don't have the thrombi, they don't destroy those red blood cells, it stopped. The complement system will not secondarily activated. And we are still working on, we try to find if there are any other examples of this sort of protective variants. And we are still thinking about maybe we can use this type of proteins that have those protective variants, and maybe they can be a good treatment target for treatment or maintenance therapy. Besides of the coagulation pathway, we also expand our ideas. We want to know if there are any other pathway involved in this disease. So I started with the known disease genes. And I queried these genes in all sorts of these databases. I try to find if there are any other similar genes, or are there any genes that interact with those known genes, or any other genes can generate similar phenotypes. I end up with thousands of results. So, and you can see these red dots are the known disease genes. Those black dots are the genes I find somehow related with those red dots, those known genes. And those black lines are their interaction relationship. Then I did an analysis called pathway analysis. Basically, it tried to find what's the general function of those black dots, all those genes I find. So not surprisingly, I found the complement and coagulation pathway because the known genes come from these two pathways. And it's easy for me to find other like interactive genes from this pathway. But a surprise to me, I find a pathway we never thought about. It's called a cell adhesion pathway. What this pathway do is they are responsible for the adhesion of the cells and also for the signaling transferring between the cells. More interesting, there's a new paper published in earlier of this year showing that cell adhesion pathway is also involved in the typical aHUS. So given this previous data, we launched a new project. We decided to screen 20,000 genes in 48 patients who is with negative genetic testing results. And we already gathered the sequencing data, and I find about 6 million variants from these patients. And my job right now is to dig into those data, find the disease variants and genes that nobody knows yet. So with those knowledge, we would better understand this disease, and we try to use this knowledge to better predict the disease risk in the families, especially for the people who carrying the same mutation but not have disease yet. And we also want to identify new therapies for long-term treatment of this disease. I would like to thank Dr. Smith to give me this great opportunity, and all the colleagues, they helped me a lot. And I especially thank the Foundation for Children with Atypical aHUS. They are sponsoring my PhD work. And finally, I thank everyone contributed to our studies and this meeting. Thank you very much.